Good morning, good afternoon, or good evening, depending on where you are. Welcome to the Virtual Journal Club, hosted by the Journal of Physiology. I'm one of the hosts. I'm Hemodynamic, a postdoc from UC Davis.
And I'm Qianwei Zhang, from PhD student from the same lab.
So we'll be hosting the Journal Club today. And before getting to the actual discussion, I would like to introduce various opportunities, especially for trainees to get involved with the Journal of Physiology. For example, we can host a future meeting of the Journal of Physiology's virtual journal club that's similar to this one. And we can write a journal club article on a recently published paper in the Journal of Physiology, or you are really encouraged to submit your own research to the journal. And the Journal of Physiology publish original research papers in all areas of physiology and pathophysiology. And if you have any questions, please email to these two email addresses.
Yeah, then I will briefly go over the structure of this journal club. So in the next 10 minutes, Haibo will give a general introduction on why this paper is relevant to the discussion. And subsequently, the editor, authors will present the paper and to give a very brief introduction about the paper we discussed today. Then subsequently, the editor will remark why this paper is accepted for publication in the journal. And then we will have around 10 minutes for discussions, and you will be able to submit your questions at any time during this session. And you can use the Q&A button at the bottom of your screen. And there will be a text box to submit your questions and autonomously. However, if you're happy to give your name and institution, please feel free to do so. Also, I would like to remind you that the session will be recorded and it will be emailed the link with the survey for your feedback in a week. And after that, there will be a 30 minutes networking session immediately afterwards. We encourage you to join this session to meet attendees and further discuss the finding in the study and the future directions. The link to the networking session will be available in the chatting box after Q&A session. So the title of this paper we will discuss today is the phenotyping heart failure using model-based analysis and physiology-informed machine learning. And being here with us today are the first authors and the senior authors of the paper. And Alice Joyce earned her Bachelor of Science in Biology with a minor in biochemistry at the University of Texas Rio Grande Valley. And she is now pursuing her PhD at the University of Michigan. And meanwhile, Dr. Benjamin Randall earned his PhD in Applied Mathematics from North Carolina State University with a minor in Physiology. And he is currently a postdoctoral fellow at the University of Michigan. And today, Dr. Benjamin Randall will join us in the panel discussion to discuss the question we may have. So please, if you have questions, comments, or remarks, you can submit it to the Q&A text box anytime, and we will discuss it shortly. Also, joining us today is the senior author of the paper, Professor Brian Carlson from the Department of Molecular and Integrative Physiology at the University of Michigan. His research focuses on the application of systematic quantitative analysis methods to understand a wide range of physiological phenomena, from smooth muscle electrophysiology to cardiovascular hemodynamics. So after my brief introduction, Professor Brian Carlson and Alice will give us a short interview or introduction of the selected paper. And we will also have Dr. Dan Beard join us in the Q&A session. And in addition to the authors of this paper, Professor Randall E.B from the Department of Pharmacology, UC Davis, who is also an editor in the Journal of Physiology, will give her remarks on the strengths of this paper and why this paper is accepted as publication in the journal. In the next 5 minutes, my colleagues Hummel will give a brief introduction on the motivation of selecting this paper to be discussed today in the journal club. Thank you.
Okay, thank you, Xianwei. So the reason for why we choose this paper really is this study is related to a serious clinical condition, which is heart failure, and often heart failure is defined as a complex clinical syndrome resulting from any structural functional impairment of ventricular filling or ejection of blood. And heart failure is associated with poor quality of life, increased premature mortality, high hospitalization rates, and substantial burden to the healthcare system. And importantly, the projected number of patients with heart failure is increasing in the next decade. And this is associated with increasing aging in the general population. And the causes or origins of heart failure is really complex and have an array of different etiologies that includes ischemic, valvular, hypertensive, or coming from primary or secondary cardiomyopathies. It can also come from congenital heart disease or pericardial disease. Despite all the different etiologies, clinically, heart failure is often described using the two types. Type 1, heart failure with reduced ejection fraction or HFrEF. And the second type is heart failure with preserved ejection fraction or HFpEF. These two types are determined by the calculated ejection fraction values. And this classification may have contributed to the variable treatment outcomes of these complex diseases. As indeed, for example, for HFrEF, we have many therapeutics that are available in order to improve the treatment outcomes. While for HFpEF, in general, there's a lack of standard and evidence-based treatment framework. And it is really crucial to identify subgroups of patients of similar physiology in order to improve the treatment. That is, in general, using a precision medicine approach. While we know this can be challenging, recently it has been increasingly recognized that by integrating mechanistic and statistical models, this will provide an excellent opportunity for achieving precision medicine goals. Indeed, starting from and integrating clinical data and coupled with mechanistic and statistical models, we can provide a better clinical interpretability and make predictions, and also to automatically gather the new metrics and identify the hidden patterns. Likewise, in the paper we are going to discuss today, that a combination of mechanistic modeling and machine learning is coupled with clinical data to provide deep phenotyping of heart failure. And I believe this is a very important step towards patient-specific treatment for heart failure. Before concluding my short introduction, I would like to also acknowledge there's a Journal Club article on this study by Troy and Loy. An interested audience should go for the Journal Club article as well. And now I'd like to with that, I'd like to pass the floor to Dr. Carlson and June for the actual presentation.
Okay, let me share my screen. Okay. So thank you for the great introduction. Can everybody see this fine? So thanks for the introduction to our paper. And before we start discussing it and tell you what we did, I want to say this is a culmination of work from a lot of different people, and we have a series of papers that have been exploring this type of methodological approach to phenotyping heart failure, but also it can be applied to actually other applications too. So Edith actually took the charge on this and did a great job. I'm going to present essentially the background, which Haibo and Jinwei have already given a little bit of background on. And then Edith's going to show you the results and essentially what we learned from what we did. So you already got a little bit of an idea here. A couple things that we wanted to point out is the exact definition of ejection fraction. When a heart fills with blood in diastole, you have to have maximum volume. And so you typically look at the left ventricle and you look at the maximum left ventricular volume. And then at each beat, a certain amount is ejected. And so that is actually the stroke volume, or the amount that ejects out divided by the end diastolic volume is the definition of ejection fraction. That has been used to create these two groups, as was mentioned earlier. But part of the reason why the treatment has been successful in HFrEF is because those HFrEF patients are very, the phenotype is very kind of monolithic. They look similar. They actually have similar characteristics in terms of not just their heart function, but their cardiovascular function as a whole. Whereas the HFpEF, there's a lot of different flavors of HFpEF. So there's many different ways to get HFpEF, and that's why we wanted to dive in and kind of explore whether we can tease out some subgroups here. So one part of how we wanted to tease this out was to use standard clinical measures. So we didn't want to actually use clinical measures that were kind of over or above what typically is gathered in the clinic, kind of these kind of special or research-oriented clinical measures, but we wanted to really say, okay, what is the typical treatment and what kind of clinical measures are typically done? And one of them is transthoracic echo, which is typically done. That obtains volume information, and it's non-invasive. That's usually the first thing that's done when a clinician suspects heart failure. You get left ventricular volumes. You can also get some left atrial volumes. We didn't necessarily use that information in this study. You also get cardiac output. You can get stroke volume and heart rate also from the volume information, from the TTE. However, you can't get any pressure information. So if the diagnosis is not really clear and the clinician wants to get a little bit more, a minimally invasive procedure called the right heart catheterization is performed, where a catheter is actually threaded into the right ventricle. It can be actually pushed up into the pulmonary artery. You can get several different right side pressures. So now we actually have in the same patient, we have pressure information and volume information. And so typically what a clinician does is they integrate these measures kind of with their experience and how they understand the heart failure diagnosis that they're prepared to apply to this patient. And so they're doing this in a qualitative manner. They're not actually graphing out a pressure volume loop, for instance, of the left ventricle. They're actually kind of understanding it based on their experience. And so we wanted to actually have more of a quantitative representation, especially in the case of heart failure with preserved ejection fraction, because there are small differences between these that need to be detected, and that necessarily can't be seen in this type of qualitative clinical approach. So this is our generic kind of workflow for this type of problem. Before I discuss it, I guess the central part of this is that we integrate these two measures together TPE and the right heart cath with a computational model. So we can actually stitch these together in a qualitative manner. But before I go through the whole workflow, I want to actually talk about the fact that we brought a sensitivity analysis forward in this procedure. So you have a computational model, you have data, and you have to understand how the parameters in this model that tell you kind of mechanistic kind of qualities of, in this case, cardiovascular function, how they can be informed by the data that you actually have. And so typically people will select parameters to optimize, and then they'll do a sensitivity analysis after the fact. We brought it front and center to kind of understand which parameters can be informed by the clinical data before we did the full optimization. And so that allowed us to say some parameters are insensitive in our model. We can fix those at nominal values, which we can calculate with kind of prior knowledge. But if we get a good estimate, fairly good estimate of that, that's good enough. And the ones that are sensitive to our clinical data, those were the ones that we actually wanted to optimize. So we took the clinical data, we optimized these sensitive parameters while fixing others. And then we got, for each patient, we got a kind of a version of the computational model, which actually represented that patient. And so we'd have, in this case, 31 different models with different parameters. From there, we can actually take these parameters, and then since we have confidence in them, we know that they're not going to be all over the place. We can actually perform some sort of analysis, and we have confidence that the output of our analysis is going to actually be reliable. And so we use unsupervised machine learning type of techniques. In this, we actually used a principal component analysis to kind of see the variance across all the patients. And then we used two different clustering techniques to actually see the groupings that might happen. And we overlaid those on this PCA analysis. And Edith will discuss this a little bit more. From there, we can actually go back and revisit the clinical data or the parameters and actually say, with these groupings now, are anything standing out that tell us the differences between one group or the other? So here's the measurements that we actually took from each one of our 31 patients. In this study, each patient is represented by a number. 1 through 10 are the HFrEF patients, and 11 through 31 are actually the HFpEF patients. One thing to note here is that the right heart catheterization and the echocardiogram are done at 2 separate times. And so quite often, if that data is available and we kind of had to kind of suss it out, there's going to be different heart rates in those 2 situations, and 2 different simulations should be run. So that's what we did in all of our cases for each one of our patients. We ran a right heart catheterization simulation and then an echo simulation and tried to fit the data in each one of those. So we've gathered all this data. Maybe we don't need to do all that work. Maybe we can actually just look at the kind of clinical data and look at the HFpEF group and see some emergent groups. But obviously, looking at this, we don't see that. The only thing we definitely see a difference in is the difference between HFrEF and HFpEF in terms of ejection fraction, because that's essentially our discriminator. But we actually don't see any obvious groups or obvious patterns that start to pop up when we're looking at the clinical data alone. So that kind of reinforced that we're going to have to dig a little bit deeper. And I'm going to hand this off to Edith, and she's going to tell you how we dug deeper.
Thank you, Brian. So yeah, so then thank you. Since we just looked at the raw clinical data and just plotted the HFrEF versus the HFpEF to see whether any patterns would show up, and as Brian said, we didn't observe anything, we proceeded to use principal component analysis, which would allow us to look at the total variance described by the clinical data that we just showed you. So in panel A, I'm showing you the results of our PCA. So in the X and Y axis are the principal components 1 and 2, which explain most of the variance in this data. So they explain 52% of the variance. Each dot represents where each patient lays in the PCA space, and next to each dot, it's the patient, the number identifying each patient. The HFrEF patients are in red, and the HFpEF patients are in blue. To provide more clarity to our PCA analysis, we decided to add a convex hull surrounding the HFrEF and HFpEF patients based on their ejection fraction classification. So one of the things we can see clearly in panel A is that the HFrEF hull and is in one end, the HFpEF is in another end, but there's a slight space where some patients overlap. So we can see that there's some HFpEF patients, for example, 11, 24, and 18 that overlap in the HFrEF space. So to test if these was based on just some shared characteristics, we decided to conduct clustering analysis. So we did this in panels B and C. We did this in K-means in panel B, hierarchical clustering in panel C. And in panel B, most of the HFrEF patients are in the K-means cluster A, and the same thing happens for the hierarchical clustering in panel C. So most of the HFrEF patients are in cluster A. So we decided to call cluster A our more HFrEF-like representative population. When we look at the HFpEF patients, most of them are in both cluster B and cluster B for both K-means and hierarchical clustering. So again, to clarify, in panels B and C, we're showing the clustering analysis superimposed on the PCA. So one of the things that we noticed is that we were not able to dissect any distinct HFpEF classifications with clustering analysis of clinical data. We noticed that there were 13 patients in the HFpEF group that consistently fell in the HFpEF PCA hole, the K-means cluster B, and the hierarchical cluster B. And then we observed that there were 8 HFpEF patients that laid in this NCC group, which we created because these were patients that did not consistently cluster. So just looking at this clinical data, we were not able to find distinct HFpEF classifications. So on to the next slide. Right. Thank you. So then where do we go from here? So we decided to, as Brian explained in our methodology, we decided to employ a cardiovascular system model shown here. And we used it's a closed-loop model, and it's an electrical circuit analogy. It's based on this, where pressure, volume, and flow are represented by voltage, charge, and current in this diagram. And it has 6 compartments representing the pulmonary circulation, the heart, and the systemic circulation. So these are the pulmonary artery and pulmonary veins, the right ventricle and left ventricle, and the systemic veins and arteries. On the right, we have table 2, which is the model parameters for our model for a healthy patient at rest. And we have a total of 16 parameters. And if we go to the next slide, we had, like Brian said, this problem of like, okay, with the data we actually have, which parameters can we inform accurately? So for this, Ben Randall, a very talented postdoc in the lab, provided his expertise in sensitivity analysis. So with a global sensitivity analysis and his help, we were able to determine which of these parameters can be identified with the given clinical patient data that we had available. So on the panel on the left, we're plotting all of the parameters and their small indices that allowed us to detect which parameters were sensitive and which were insensitive. And we ended up selecting 9 parameters to optimize, highlighted in green here and also below for each patient. So next, I want to show you an example of our model predictions. So on the left, we have a HFrEF and a HFpEF on the right. And this is just a representation of what our model outputs are. So in red, the dashed lines represent the clinical data, and the smooth lines represent our model simulations. So in panels A and E, respectively, we're looking at the pressure data in the left ventricle as well as the systemic circulation. Panels B and F represent the pulmonary pressure as well as the right ventricular pressures. In panel C and G, we're looking at the volumes in the left and right ventricle. And panels D and H is an integration of a PV loop based on our simulations and also making sure our simulations are matching the actual patient data. And this is what gives us confidence on the hemodynamic parameters derived from our model. So one of the things to note from this representative patients is that, for example, HFrEF patients, they have higher volumes than HFpEF patients. So this is something that we consistently observed. So onto the next slide, I can show you that we decided to run our hemodynamic parameters describing the state of each patient through our workflow using PCA, k-means, and hierarchical clustering. And in panel A, I'm showing you again a PCA of this data, of the hemodynamic data for each patients. And we noticed that, there was like a very clear overlap of HFpEF patients 29, 18, 17, 11, and 30 in the same space as the HFrEF population. And again, we had this question of, is this based on physiologically shared characteristics among these HFpEF population with the HFrEF? So we employed k-means and hierarchical clustering, and indeed we observed that for both k-means and hierarchical clustering, these HFpEF populations always align the same grouping with the HFrEF. So we created this HFrEF-like HFpEF group called HFpEF1. And again, these are 5 patients overlapping in the same PCA region as the HFrEF, k-means, and hierarchical clustering. We also observed that there were some HFpEF patients that always cluster with HFpEFs, and they were always in the same PCA space as most HFpEFs. So we call this our HFpEF2 group, which is our classical HFpEF population. We also found a subpopulation of HFpEFs that we called NCC that did not consistently cluster among these techniques. So then onto the next slide, I can show you what we did. We actually decided to plot the optimized parameters or hemodynamic parameters describing the physiology of these patients based on these distinct heart failure subgroups, specifically HFpEF. And we saw some interesting things. So panels A through E represent what this infographic table is showing. So the most relevant parameters. And we found something really interesting. So the main parameter driving HFrEF was a decreased level in left ventricular active contractility at rest compared to our normal human, our healthy normal. So this cardiac parameter is the main thing driving this HFrEF population. The other thing we noticed is that the HFpEF1 was very similar to the HFrEF population with very similar parameters and hemodynamics driving this phenotype. The other thing we noticed is that our HFpEF2 population had not only cardiac dysfunction, but also a lot of issues with the systemic and pulmonary circulations with increased levels in their both systemic and pulmonary resistances. They had very increased left ventricular passive stiffness at rest, indicating that, they had very high just diastolic dysfunction in accordance with our classical understanding of HFpEF. So this is indeed a very classic HFpEF group. Our NCC population is almost in between these two groups, so which lets us to believe that NCC could be its own HFpEF population, or maybe a classic HFpEF group that's transitioning into a more HFrEF-like state. Again, we decided to, on the next slide, we can show you that, we decided to actually go back to the clinical data and see whether any distinct things will show up based on this new group. And indeed, we found something really interesting. We found that if we just look at panel A, as we expect, the ejection fraction for HFrEF is lower than that of like the HFpEF groups, but we did observe some distinct differences in even ejection fraction in the HFpEF populations. And if we dig deeper, we can see that in panels B and C, we have plotted here the systolic and diastolic left ventricular volumes. We see some trends. So we observe that the HFpEF1 group has higher systolic and diastolic ventricular volume as in HFrEF. Our HFpEF2 and NCC groups have lower systolic and diastolic ventricular volumes, in accordance to the more classic understanding of HFpEF. So onto the next slide, I can show you that So I forgot to mention it, but yeah, so these knowledge about the volumes allowed us to, just maybe think that these could be left ventricular volumes could be an indicator of these distinct groups, but we couldn't have been able to get to that conclusion without our methodology using physiology-informed machine learning. And yeah, so one of the nice things is that recent studies have determined HFpEF subgroups using machine learning, but coupled with non-classical, just routine clinical measures. So we have a group that did RNA sequencing in endomyocardial biopsies. They found 3 distinct HFpEF groups, one of them very similar to HFrEF. We had another group did extensive quantitative echocardiography. They went above and beyond. Just having very rich insight into echo data. They also found 3 distinct groups, and then another group also found plasma protein profiling using that, 3 distinct phenotypes of HFpEF. And these studies are really interesting, and it's nice to see that other groups are also finding these distinct classifications. I think one of the things that we were really happy about our study is that we were able to use just routine clinical data that would be used either to diagnose or keep treating heart failure patients to arrive to these conclusions. And we hope that it could be of use in the clinic. So now after reading this work and this paper, I'd like you to leave with these key points and conclusion. We found 3 subgroups of HFpEF emerging from our deep phenotyping approach using cardiovascular model and informed machine learning. A HFrEF-like HFpEF group, a classic HFpEF group, and a group that exhibits characteristics of both. Our methodology reveals that potential indicators for identifying HFrEF-like HFpEF patients are elevated left ventricular systolic and diastolic volumes. These differences in determining HFpEF subgroups could not be distinguished based on the clinical data alone. It was after we used our methodology, although we wouldn't have gotten here without the clinical data to parameterize our model specifically to each patient. And ultimately, the combination of mathematical modeling analysis, machine learning techniques provides unique insight into the classification of HFpEF. With that, I'd like to just acknowledge all the great people that contributed to this work. Benjamin Randall, he contributed so much of insight into this work, as well as in his expertise in the sensitivity analysis. Scott Hummel and David Kamener were our cardiologist liaisons, just providing so much insight into understanding the clinical data, the value for clinicians, and also analyzing the echo records. Daniel E.B and Brian Carlson for belonging to their lab and envisioning and steering this project. We'll also like to thank our funding sources and the Journal of Physiology Club for hosting us today. Thank you.
Yeah, thank you very much for your very insightful presentation. And I think finding here is quite interesting. So before reading some questions for Q&A session, let me invite accepted for publication in the journal. So just your questions in the Q&A column, and we will discuss later. Thank you. So Professor, are you ready? Thank you for reading.
Thank you, Jean-Wei. I would also like to thank Haibo and Jean-Wei for organizing this virtual journal club, and job very well done, Edith and Brian, for presenting the main methodology and results, and congratulations to all of the authors. I'm Ele Grandi. I'm one of the reviewing editors for the Journal of Physiology, and I was a reviewing editor for this paper. So I'm very excited to see it presented. My job today is to kind of give our audience an idea of why we thought this paper was suitable, and why it was accepted for publication in the Journal of Physiology. So I'm gonna kind of restate some of the things that have been already said. This manuscript really illustrates a powerful approach in combining patient data that, as we heard are obtained through routine clinical measures, mechanistic mathematical modeling, and statistical learning to add new insight into the classification of heart failure. And we consider it a little bit of an icebreaker for this approach in the cardiovascular field. But to our knowledge, this is really the first paper to define the phenotypic landscape of HFpEF using these patient-specific mechanistic models to enrich clinical observations. We also think that this approach might actually contribute down the line to informed treatment, which is really badly needed for HFpEF that really lacks any effective treatment. And as also Brian alluded to at the beginning of his presentation, we really think this approach is of general interest and can have potentially much broader applicability in other fields of physiology and pathophysiology. One last thing that I want to mention is also the rigor in this paper. So as a computational model, I was happy to see how the authors really carefully constructed their models. And this also means that they very clearly clarified their assumption. Brian also alluded to this idea of using sensitivity analysis upfront to kind of decide which are the parameters that are even worth that can be identified because they are sensitive, and as well as I think part of the rigor is also in acknowledging the limitations of the approach. So all in all, I think I will repeat this. I think this is really an icebreaker paper that will probably open up new ways in which we look at classification of heart failure patients, and as we say, could inform treatment and could be more broadly applicable to other pathophysiologies. And I think I give it back to Haibo and Zhongwei to moderate the discussion. Thank you.
Thank you. Thank you, Professor Grandi and Professor Carlson and Edith for the insightful talk and remark on the paper. So the Journal Club now is open for questions. And while we have one question here, everybody is encouraged to put your question in the QA box or in the chat box if you prefer. So now we have a first question from Linda Blinsky. Is there any connection between the RNA plasma membrane and other non-standard clinical measures to which machine learning have been applied, and the most important parameters in the model for distinguishing the HFrEF and HFpEF phenotypes?
So, so one of the things is that in our study we didn't look at the RNA, plasma protein, and the other non-standard measurements. So it's a little bit hard to compare to those, but we did observe some similarities between the groups. For example, there was one group that they were looking at one HFpEF population that had the highest stiffness. And looking at some of the descriptions, it looked like it was similar, for example, to our HFpEF2 group, which was the classic HFpEF. Then looking at the group studying RNA sequencing in the biopsies, there was a population that was very similar to the HFrEF. But because these studies didn't look at hemodynamics, it's a little bit hard for us to extrapolate. We're currently trying to work on analyzing some data that would allow us to provide more insight into the actual biochemistry of these distinct groups.
Okay, and also thanks for answering. And we also have another questions pop in the chatting box. So from Claire Owen. So great overview of your work and thank you. I'm a cardiac physiologist and working on my PhD in orthostatic hypertension. My research question is to use cluster analysis to find patterns in blood pressure profiles. So my question is, did you find any pushback or challenge on the small sample size using machine learning techniques? Okay, that's also my what the question I want to ask. Thanks.
Well, so yeah, there was limitation on the machine learning techniques we could use. So for example, we couldn't do things like neural networks or just more advanced things that would require, a training set and a testing set. So that's where in following, actually, we're currently working in expanding our data pool to be able to do more advanced studies. So that's, and also we want to work on just keep validating these results because we did just use 31 patients. Yeah, I mean, we were able to implement at least these, PCA, k-means and hierarchical clustering fairly easily, even with this small population size. I don't know if Brian, you want to add something to that?
Oh, no, I think that's a great summary. And yet, really, this is kind of almost a proof of concept. But you're right, really expanding not just the HFpEF population in terms of the patients that we have, but also looking at other types of heart failure, HFrEF and maybe dilated cardiomyopathies. Where do they sit in this overall space? Will be informative. So as we get larger population, we can start to relate this HFpEF population might actually be similar to, maybe there's a group that have diabetes that is kind of part of this month, but obviously that has to come by expanding this number. A lot of hospitals around the country and a lot of medical kind of university hospitals are starting to gather large data sets. And so there's actually a large, it's called DOCTOR, the Data Office of Clinical and Translational Research here has a large database that goes back to early 2000s, which with millions of patients in there. And now we can actually pull, so it looks like we can pull around 800 patients that have an echo right heart cath and are diagnosed as having diastolic dysfunction. So that will be somewhere down the road to see if what we showed here actually stands.
Great. So we have another question coming from Zoe Adams. The question is, did you control for comorbidities or different medications in your model?
No, we decided to just walk in blind, just HFrEF, HFpEF, and see what we found. But it'll be interesting to go back and see what are the comorbidities that the HFpEF in the distinct groups have, or on what medications were there, to see if there were any similarities and if they got better or not.
There was one interesting thing in our initial procedure. We actually had a larger population of patients, and after doing some of the analysis, Edith said to Scott Hummel, this patient looks kind of strange. This doesn't look like a HFpEF patient at all. And Scott looked this patient up, and it ended up that this patient did have had cancer, and so they were on some cancer drug, which actually moved them from HFpEF to some other space, right? It was more HFrEF-like, but Edith was able to see that in our initial analysis. So there's some hope that when we do factor in some of that, and again, this is a small size, so we can't really say much about that, but small sample size. But I think some of these things will emerge. We just have to be selective about what we look for, and otherwise, everybody would be a different cluster, right?
Yeah, great. So I have a question here. I was wondering, your approach can be helpful to determine the mechanisms underlying sex differences. Obviously, we know there are sex differences that exist in both the symptoms and treatment outcomes, for example, in HFpEF. And can your model help to determine why there or where these differences come from, and how we can can be helpful to determine the better therapeutics?
Yeah, I think it can be helpful. So, for example, we observed that there were some, like the HFpEF group that's similar to HFrEF. So potentially therapeutics that are successful for HFrEF could be applied to that group. especially to improve their left ventricular active contractility. We also observed that the classic HFpEF patient has a lot of issues with increased resistances in both systemic and pulmonary circulation. So maybe addressing those aspects could help the load of the heart in that patient subpopulation. And yeah, it would be really interesting to detect if any sex differences show up. I think it was the study by Cohen et al. That they found that one of their groups was consistently of, the majority was elderly women that had that specific heart failure HFpEF phenotype. So it'll be really interesting as we expand our HFpEF pool to see what more clear phenotypes appear and also how they correlate more with like these other studies. Did I answer your question?
Yeah.
Thank you.
And we had,
I was just going to say, yeah, that's a great thing to look into. So, I mean, we didn't necessarily look at that, but we could go back and we'll probably do that later this week.
And we have another question from the audience. Mohammed Aliyu, sorry, I'm pronouncing your name badly. Mohammed Aliyu Uma. How variant is the blood pressure of HFpEF and that of HFrEF?
So what I'm understanding is how variant is the pressure, the blood pressure for HFpEF and HFrEF. So usually they're both pretty high, but usually HFpEF patients have higher pressures than the HFrEF population.
Okay, I'd like to ask the last question before we closing and moving on to the network session. The question is related to the sodium glucose transport protein inhibitors that recently been identified to be really helpful for both types of heart failure. So, and there are many ongoing studies to identify the mechanism why this is really helpful and better than other drugs. So would your approach be helpful to determine the actual mechanisms behind the treatment outcomes of this particular new class of drugs?
So yeah, it will be really interesting to see. You get, for example, like the clinical measurements for those studies, and like if they have pressure and volume data, it will be really interesting to run it through our group and see what happens with the physiological state of each patient. What parameters get better. Yeah, so like for both HFrEF and HFpEF. That would be like something interesting that we could do, and I think I hope that's one of what happens with this work, that people apply it for their distinct clinical studies and see and can test what aspects are getting better for a specific patient population.
So I would add to that, Edith, that even like a simpler, I think your answer is a very good answer, but I think a simpler way would be, can we, how do responders versus non-responders or different degrees of responsiveness, phenotype before drug treatment, right? And that's probably likely to give us clues about the mechanism that's being targeted, given that, this sort of phenotype is a responder or a better responder than that sort of phenotype, right? Or that would be the hope, a hope.
Great, thank you. Thank you very much. I believe we are approaching the end of the journal club, and we now have a link for the network session, the posting in the chat box.